are a myriad of RNA binding proteins that are related to the hematological malignancies, with leukemia, lymphoma, myeloma, or dysplasias. For example, in leukemia, the Musashi proteins, WT1, or the molecule that is our focus of our research, the hnRPK, contributes to the development of acute myeloid leukemia. Indeed, the subtype of 9q deletion acute myeloid leukemia have a deleted hnRPK protein that produces a haploinsufficiency of hnRPK and drives the acute myeloid leukemia. This contrasts with the role of the hnRPK in the lymphoma, in the DLBCL, that we see that the hnRPK is overexpressed in DLBCL and drives the disease and correlates with the poor prognosis of the disease. Interestingly, our last findings through the development of a hnRPK overexpression widespread mice model show that the hnRPK overexpression promotes the hematopoietic stem cell exhaustion, promoting a dysplasia phenotype, a bone marrow failure. The molecular mechanism of this bone marrow failure is because hnRPK promotes the cell cycle arrest and the senescence phenotype, and we think that is related with the nuclear stress that produce the hnRPs like hnRPK. The nuclear stress promotes dysfunction in ribosome. In our case, the dysfunction is a enhancing of activity. That is a bit weird because the most common phenotype, molecular mechanism in the bone marrow failures, like the ribosomopathies, are a dysfunction in a failure of the function of the ribosome, but our phenotype is completely the opposite. We observe an increase of the activity of the ribosome. And as I said, that promotes the cell cycle arrest and the senescence phenotype and the bone marrow failure phenotype, but also aging phenotypes like kyphosis and other signs and symptoms. That is interesting because when that produced in hematopoietic stem cells, produce bone marrow failure, but when happens in a cancer cell, like a acute leukemia cancer cell, or a lymphoma cancer cell, or a myeloma cancer cell, promotes the resistance to any source of stress, like the treatments produce. Because a senescent cell is by definition a resistant cell to the treatments. That causes that the hnRPK overexpression, but other RBPs overexpression is correlated with a poor prognosis and poor outcomes in a plethora of hematological malignancies like leukemia, lymphoma, and myeloma. We are studying how to overcome this resistance, targeting directly hnRPK, but also targeting the functions of the hnRPK and other RNA binding proteins, like the ribosome function through, for example, MYC or mTOR pathway, the senescence phenotype through senolytics, and the nuclear stress to try to overcome this resistance and get the successful treatment of the frontline treatments of myeloma, acute leukemia, and other hematological malignancies.